In this video, you will learn how to prepare, run, and interpret a gel. First, make 30 milliliters of agarose. For this video, we made agarose that is 1x TBE and 0.75% agarose. To do this, combine 3 milliliters of 10x TBE buffer to 27 milliliters of water, thus diluting it to 1x TBE. To this, add 0.225 grams of agarose powder. These ratios may be changed to reach the desired consistency of your gel. Heat the agarose mixture in the microwave for 1 minute in a plastic container. Carefully remove the container from the microwave. Add 1 to 2 microliters of gel green, which will aid in visibility after the gel is run. Make sure to leave the tube of gel green in the dark at all times it is not in use. Leave the gel to cool for 10 to 15 minutes. Do not pour it while it is still hot. Pour the gel mixture evenly into both halves of the gel mold. Even though this procedure makes 2 gels, you will only use 1. Add the comb to the filled gel mold. The smaller side is preferred, but you can use the larger side as well. Leave the gel to set for 15 to 20 minutes. When the gel is set, gently remove the comb and remove 1 gel from the mold, making sure not to break it or get fingerprints on the surface. Place the gel in the gel rig, making sure to align the wells with the well marks in the rig. Make 150 milliliters of 1x TBE running buffer. To do this, combine 15 milliliters of 10x TBE buffer to 135 milliliters of water. Pour the buffer into the gel rig, making sure it covers the surface of the gel. Add 1 microliter of purple loading dye to every 5 microliters of product. This will weigh down your product and keep it in the wells. Add 5 microliters of DNA ladder to the first well of the gel, adding 5 microliters of each product into the following wells. Make sure the filled wells are right next to each other and do not leave empty wells between them. Turning the blue light on while doing this can help with visibility. Make sure the filled wells are right next to each other and do not leave empty wells between them. Place the cap on the gel rig, turn off the blue light, and press the power button. Allow the gel to run for 20 minutes. Before interpreting the gel image, it is beneficial to draw out what you expect it to look like. This image shows a gel prediction beside the resulting gel. The comparison of the two makes discrepancies more obvious. To do this, you must know the size of your parts, including how they have been digested and the size of the backbone. The smaller a part is, the further down the gel it will run. This gel image includes from left to right a 1 kb ladder, a digested product with 2 ligated parts in a backbone, a digestion of 1 part in a backbone, and a key for the bands of the ladder. By referencing the ladder, the approximate size of the part can be found. A chloramphenicol backbone, which is about 2070 base pairs, appears in both parts. The lower band in the second column is a ligated part at around 1800 base pairs. The lower band in the third column is 1 part of the ligation at around 1000 base pairs. This analysis can be used to determine if a ligation was successful and if the expected parts were present.